Good morning, children. Up to now, we are doing the questionnaire part of the lesson Nutrition in Plants, and we'll continue it now. Okay. So we have dealt with the conceptual understanding based questions and different questions about the asking questions and making hypothesis, isn't it? Now we'll move to the experimental basis question, that is the type of how to do this or prove the presence of with different items in the lesson that are dealt in this lesson. Okay, children. So now we are seeing about the essential necessity of the light for the preparation of food in the plants, that is photosynthesis. Okay, so see the question, children. How do you prove light is essential for photosynthesis? You know this, I have already told you in the lesson how we'll prove this. So how to write the answer for this question? For this you have to divide the answer into four parts. About first, aim. What is the aim of the question? That is, light is essential for photosynthesis. Next step is materials required, that is apparatus. So you have to write what are the different materials required for to doing this experiment. Next, procedure or the experimentation basis of what to do in this experiment, that will rise step by step. So how do you do this experiment? Next, you have to do observe what happens in the experiment. Later you have to give the result or the inference. So in these 5 steps you have to give the answer for this questions. That's how you have to write this question. See the answer here, children. Aim: to prove light is essential for photosynthesis. Apparatus: 4 to 5 leaves of hibiscus plant, black paper, iodine and alcohol are taken. Experiment: take 4 to 5 leaves of hibiscus plant covered with black paper without removing them from the plant. The way the black paper was cut and fixed to the leaves is shown in the figure that you have to show. See here. So this is the figure you have to draw neatly, children. So like this you have to draw and you have to answer this question. Okay. Next. These leaves were plucked 2 days later. Their green color was removed by using alcohol solution and were dipped in iodine solution. Write down this part and we'll write about the observation part. Note it down. So in this experiment, the plants are completely kept under the sunlight, isn't it? So while observation of taking the leaves out of the plant, and the leaves do not turn into blue or black color in the part of the leaf which is covered by black paper, and other parts turn blue. Is it clear? So the leaves do not turn into the blue or black in the part of the leaf which is covered by black paper, but other parts turn blue. Here, right. Okay, the result. So the parts which are turned blue says that it contains starch, and the part that doesn't turn blue says that it doesn't contain starch or they did not prepare the food. So starch is not produced in the leaf part covered with the black paper, and starch is produced in the leaf part which is covered with black paper in the presence of sunlight only. Okay, children. So this experiment confirms that light is essential for photosynthesis, that is for the preparation of food. The plant needs sunlight. Is it clear? So that is the experiment. In the second question, we'll prove that the next question is about the why are we heating the leaf, that is boiling the leaf in alcohol or hot water. What is the reason? In this experiment we did it, isn't it? So in the carbohydrate test of the plants, we are boiling the leaves in hot water or in alcohol. Why is it so? Because the green color disguises the blue color and the test cannot be confirmed easily. So we are boiling the leaf in the alcohol solution or in the hot water. That is the simplest form of question that we have to reason it. Okay, so that is the reasoning question we will write in the next part. You do up till then you write this question. See the question, children. Examine the experiment of carbohydrates present in the leaves. Why is the leaf boiled in alcohol? So answer is, in the carbohydrate test with iodine solution, when the leaf is put on the leaf or the leaf is put in the iodine solution, it should turn to blue if the starch is present. The green color of the leaf disguises the blue color. So we must first remove the green color of the leaf, and if we want to test for the starch, then only we can identify the presence of starch in the leaf or not. The way to do this is to first put the leaves in a boiling tube and boil them in alcohol or hot water. Is it clear, children? So after this question, we'll move to the textual questions. Okay, children? Till then you write this answer. So this is the question that is related to the light experiment that you have done previously. Light is essential for photosynthesis. Take your textbook, page number 102, children, and the questions, improve your learning questions are being given here. You can see the first question here. What is there? It is given a potted plant kept in sunlight responds to starch test and the part for to 1 day or for 1 day even if it responds for the starch test, but the same potted plant when it is kept in the dark for 2 days doesn't respond to starch test. What is the difference? Why is it so? What is the reason for this? They have asked the question. So here, will there be the difference in the two potted plants kept in dark and light? Yes, there will be a difference. You have to write. So when the leaves of the potted plant kept in a day for 1 day and the leaves of the potted plant kept in the dark for 2 days, when they are tested with starch, there will be the difference. What is the difference? The leaves of the plant which is kept in light for a day responds to starch. The same plant when it is kept in dark for 2 days does not respond to starch test. Starch test is we do with the iodine solution. If it turns to black or blue, it says that it contains carbohydrate or starch in them. If it doesn't, it does not contain carbohydrate or starch in them. So that is the test we are writing here. And the reason, as the sunlight is not available for the plant that is kept in darkness, there is no available formation of starch. Okay, and hence the plant does not respond to starch test. This is the answer. Hope you have understood this question, children. And next we'll move to the second question of your textbook. You note down the question by seeing the textbook and write the answer, children. Okay. Next, second question of your textbook. What happens if the leaves of the green plant are coated with oil? If the leaves of green plant are coated with oil, the stomata present on the leaves gets closed. So, what happens? The main functions of stomata are photosynthesis, respiration and transpiration. All these functions cannot take place in the leaves. So, the plant dies soon. So, that is the answer for this question. Okay, children? So, in this manner, you have to write the answer. If the leaves of green plant are coated with oil, the stomata of the leaves get closed. There will be no gaseous exchange with affect the rate of photosynthesis. There will be no transpiration too. The plant dies soon. Right, Sunma? Next question, children. Do you think saprophytes clean up or help us in keeping the environment clean? You know what are saprophytes? Saprophytes are the organisms which depend on dead and decaying organic matter. So when they depend on it, they clean the earth's surface by feeding on it. Okay, so we can say that these help us in keeping the environment clean by feeding themselves on dead and decaying organic matter, and thus they help in cleaning the earth's surface. That you write down the answer. Next we move to the differences between the parasites, saprophytes and also the symbionts, everything. Okay, move it. Write down. The third question, next one here we have completed the third question. Now the differences part. Parasite and saprophyte and host and parasite. We taken first parasite and saprophyte. Parasite, they depend on host plant for their food and they get the food from their host itself. They live in or on the host plants. These plants do not have chlorophyll, neither chlorophyll nor leaves or chlorophyll. So here, especially parasite example cuscuta. You also have the parasite in the animals like malarial parasites, roundworms also. They are parasites which live in another organism. But here we are dealing with the nutrition in plants. I am giving the example of only the plants, and the other ones also I have told you while I am explaining the lesson about Urabanke and all, isn't it? You can write down those examples also. Saprophyte. These organisms live on dead and decaying matter and get their food from the dead and decaying matter in itself. These plants also do not possess chlorophyll. So these examples are fungus, that they belong to fungi family, and it is mushroom is an example of it. Note down this. Next we will move to host and parasite. So here the difference between host and parasite. Host, especially the host, is a green, in plants if you take, it's a green plant which can prepare their own food. Often animals also become the host for other animals, like human beings also become host for the other insects and other animals, that is a small microorganisms. So here host prepares its own food. For example, in plants, it is green plants as they prepare their own foods in the presence of chlorophyll because they contain green leaves which prepare their food containing the chlorophyll in the presence of sunlight. Next, parasite. Already I have explained you before about the parasite that they depend on the host plant for food, and they live in or on the bodies of the host plant and survive over them.